All right, welcome back, fellow scientists. So we're going to continue our discussion of biotechnology, which, if you'll remember from the last video, is manipulating DNA to benefit humans. And we're going to look at two tools and we're going to look at two processes today. So if you remember, our first tool was restriction enzyme from last video. Our first process was transformation. So our second tool of biotechnology is something called PCR. PCR stands for polymerase chain reaction. So basically what PCR does, you can you're familiar with the word polymerase because we talked about DNA polymerase, which is active in DNA replication, and RNA polymerase, which is active in transcription. So it's that same enzyme, it's that same polymerase enzyme which makes more DNA. And so what we do is, let's say that we have a sample of DNA, and we want more of it because we want to play around with it. We want to study it, right? So what we're going to do is we're going to put it in this PCR machine, and what the PCR machine does is it takes this sample of DNA and it heats it up. So it heats it up, and what the heating does is that separates the two strands of DNA. It breaks the hydrogen bonds and it separates the strands of DNA. Then polymerase will come along and it will do what it always does and add nucleotides, complementary base pairs, A's with T's, C's with G's, just like that. And so now we have two strands. But it'll do this like thousands of times. So it'll heat it up again, and so then we'll have, oh boy, this is going to get complicated. So then we'll have that strand, and we'll have that strand, and then we'll have that strand and that strand, and then it'll let DNA polymerase come through and add nucleotides like that, A's and T's, C's and G's, just like that. So now we started with one double helix, and now we have 4 double helices. So like I said, it'll do this hundreds if not thousands of times until we have millions or billions of copies of the original DNA, which these millions or billions of copies, because we have our base pairing rules and because we're using DNA polymerase, it's the exact same as this first copy. So that's our that's our second tool of biotechnology, is polymerase chain reaction. Our third tool that we're going to talk about of biotechnology is called gel electrophoresis. Our third tool is called gel electrophoresis. And so the basic process of gel electrophoresis separates molecules based on size and charge. Based on size and charge. So this is how it works, and this is actually something that we're going to do next week, borrowing equipment from Whitworth, and it'll be really cool. So our first process is to make a gel. Or our first step in doing gel electrophoresis is to make a gel. And this gel, and we'll see there's 2 different kinds of gels that we could make. We could put little indentations, or they're called wells, in the gel to one side, or like we're going to do in class, we could put the wells right in the middle, the little indentations or the little holes or the little divots, the little wells, just like they talk about back in Bible times. You dig a well, you dig down, right? So if we look at the gel from the side, then these little wells are just little indentations. This is looking at the gel like it's laying on the table, top down, and then this is looking at the gel like you're eye level with the table, like you're looking across the table. And this is the top of the gel right here, and this is the bottom of the gel right here. So these wells are just indentations in the gel. And the gel is just, I mean, it's basically, it's like the agar that we put in the petri dishes. It's like Jello. In fact, we use agar, and we use, I mean, the same stuff that we use to make Jello, we use with the gel. So our first step is to make the gel. Second step, you put it in a special chamber with water. So chamber and water. Our third step is to put whatever we want to separate into the wells. So put material into the wells. And then we run an electric current through the chamber, through the water. So we apply an electric current to the chamber. So that's our fourth step. Apply electric current to the chamber. And what that does, so let's say that we have a molecule here. Let's say that we have our negative side connected over here and our positive side connected over here. Let's say that in this well, we've put a positive molecule. Right, so that positive molecule, opposites attract, so that positive molecule is going to go this way. Now let's say that in this well we put a negative molecule. Well, that negative molecule is going to want to go this way, right? So we're separating molecules based on size and charge. And in fact, we can have two different molecules. We can have a positive and a negative in the same well. And this is why gel electrophoresis is so powerful, right? The positive molecule is going to go this way, and the negative molecule is going to go this way. So we could have one sample, and we could use that to separate or sift or strain the positive molecule away from the negative molecule. Okay, so that's the first thing. Now what we can also do, and again, let's say this is negative and this is positive, we also separate molecules based on size, right? So let's say that we have all negative molecules in these wells, and let's say we have a couple different negative molecules. Some are bigger, some are smaller. So because this is a gel, the larger molecules are going to move slower through the gel, and the smaller molecules are going to move faster. So that as these negative charges migrate or move through the gel towards the positive end, these down here towards the bottom of the gel, these are going to be smaller. And these up here towards that stay kind of towards the wells, these are going to be larger. So that's how we separate molecules based on size and charge. And that's our third tool of biotechnology, gel electrophoresis. Now, these two processes go hand in hand. So our second process of biotechnology, remember our first process was transformation. Our second process is DNA sequencing. And I'm not going to go very in depth. There's other really good videos out there that describe this process in depth. But basically, we can use the combination of PCR and gel electrophoresis, and we can determine the exact order of A's and T's and C's and G's in a piece of DNA. So DNA sequencing is determining the order of nucleotides. So A's and T's and C's and G's in a sample of DNA. So that's DNA sequencing. And oh, by the way, we have sequenced the human genome. I believe that was done in 2003. I think it was announced in 2001 and then officially completed in 2003. But we now have a catalog of all the A's and T's and C's and G's that we need to make a human. So that's pretty cool. So that's the second process of biotechnology, DNA sequencing. And then a very similar process, but slightly different, slightly less complicated. Our third process of biotechnology is DNA fingerprinting. And so this is less specific than DNA sequencing. DNA fingerprinting, we're using that because each person has their own unique individual DNA, except for identical twins, of course. We can use DNA fingerprinting to identify a person's DNA, or identify a person and attach that to a DNA. So DNA fingerprinting is identifying a person identifying a person based on their DNA. And again, this is a more in-depth process, and there's a lot of other videos out there that can explain it a lot better than I can. So we'll just leave it at that. That DNA fingerprinting is an identifying a person based on their DNA. So this could be used to determine paternity or maternity. There was a case with a tsunami in, I believe it was Indonesia, where a baby was washed out to sea and they survived, and so they showed up like 70 miles down the beach and they didn't know who their parents were. And so they used DNA fingerprinting to identify, okay, these are the parents of this baby, because there were lots of people who lost tiny babies, and so there were lots of people who thought that that baby was theirs. We could also use it when investigating a crime scene. If a perpetrator leaves DNA evidence, such as like hair or skin or something like that, at a crime scene, then we can analyze that DNA and we can compare it to suspects of that crime. Now, caveat, just because a suspect's DNA matches the DNA at the crime scene doesn't necessarily mean that they committed the crime. It just means that they were at the crime scene. At some point in time, not necessarily when the crime was committed. So anyways, DNA fingerprinting is not cold, hard, yes, this person did it, but it's just another piece of evidence that lawyers can use to try to figure out the truth of what actually happened. So that's our second and third tool of biotechnology, polymerase chain reaction and gel electrophoresis. Those are our two tools that we talked about. And then our second and third process, DNA sequencing and DNA fingerprinting.